So this figure here shows an excitatory pathway from the prefrontal cortex down to the amygdala. And by excitatory, what this means is that it's more likely to make the next structure fire an action potential. So the amygdala contains GABAergic interneurons. So interneurons, well, inter just means that they send within one brain region. So it contains GABAergic interneurons. So the amygdala basically has GABAergic interneurons, but it also has glutamatergic projection neurons. The amygdala has GABAergic interneurons, but it also has projection neurons, and projection neurons project to a different part of the brain, so a different region, different structure of the brain, and these are glutamatergic. So GABA is the main inhibitory neurotransmitter and glutamate is the main excitatory neurotransmitter. So the GABAergic interneurons, what they do is they inhibit the glutamatergic projection neurons. So if these GABAergic interneurons are under control, what they're going to do is they're going to regulate the projection neurons that are glutamatergic from being sent. And the GABAergic interneurons, this modulation will decrease this activity of the glutamatergic projection neurons being sent out from the amygdala. So this excitatory pathway here in the prefrontal cortex that sends to the amygdala, in schizophrenia spectrum disorders, it's thought that this excitatory, so this glutamatergic pathway, may fail to basically modulate, and that means increase these interneurons in the amygdala. If this glutamatergic pathway in the prefrontal cortex to the amygdala is not increasing the GABAergic interneurons of the amygdala, they're not going to modulate the glutamatergic projection neurons here. So this isn't going to happen in schizophrenia spectrum disorders, which means that these GABAergic interneurons are not stopping the projection neurons from firing. So the glutamatergic projection neurons in the amygdala are going to become overactive. So there's also a glutamatergic pathway from the amygdala to the hippocampus. So this is excitatory. In schizophrenia spectrum disorders, the projections from the amygdala, because the amygdala is overactive, so that's overactive, this is sending more glutamatergic projections from the amygdala to the hippocampus. And what this can do is this can damage the hippocampus. The hippocampus becomes damaged. So the hippocampus damage can basically mean that this is one of the reasons to why there is reduction in the size of the hippocampus. So it's often said that there is a association between increased lateral ventricle size and decreased hippocampal volume, and this could be one of the reasons why the hippocampal volume is decreased. There's also the theory that the increase in lateral ventricle size kind of pushes and reduces the hippocampal volume, but this is one of the reasons to why the hippocampal volume may be decreased, is because the hippocampus is being damaged by the overactive amygdala. So far the prefrontal cortex fails to modulate the amygdala, so the amygdala is then overactive, and that sends the hippocampus, and that's glutamatergic, that results in damage to the hippocampus. So the hippocampus also sends glutamatergic projections to a load of other brain structures, and the excitability of those structures is modulated by the interneurons in the hippocampus. So the hippocampus, so I just put that there, has interneurons that are GABAergic. And one of the structures which is shown in this diagram here, the hippocampus projects to, is the nucleus accumbens. So if the GABAergic interneurons in the hippocampus are damaged, such as those that have schizophrenia spectrum disorders, so the excitability of the glutamatergic projections, so those excitable projections to the nucleus accumbens can't be controlled. So the amygdala, the GABA neurons do not modulate the glutamate, which means that it's overactive. The overactive amygdala project to the hippocampus. So the hippocampus also has GABAergic interneurons, and because they're damaged, that's also sending out more projections. So the same within the amygdala, how the GABAergic neurons are failed to modulate the glutamatergic projections, that causes then the hippocampus damage to their GABAergic interneurons, which failed to modulate their excitable glutamatergic projection neurons. So because this projects to a load of other brain structures such as the nucleus accumbens, that overactive hippocampus, so the overactive hippocampus, what that's going to do is going to lead to hyperactivity in some of these other brain areas such as the nucleus accumbens. So the nucleus accumbens contains something called medium spiny neurons, MSNs, and what they are is GABAergic projection neurons. So the GABAergic projection neurons, and one structure that they project to is called the VP, the ventral pallidum, and this is an inhibitory pathway. So because these project here and these are GABAergic, this is going to inhibit the firing rate of the ventral pallidum. So because this is hyperactive, this ventral pallidum is going to be decreased in its activity because these GABAergic neurons are firing more, and more GABA means more inhibition. So the VP is inhibited. And in the VP, similar to the nucleus accumbens, the VP also has GABAergic neurons. So the ventral pallidum is being inhibited by the nucleus accumbens. And similar to the nucleus accumbens, the ventral pallidum also has GABA neurons. And some of these neurons project to the VTA, to the ventral tegmental area. And what they would do is inhibit this area. So GABA should inhibit the VTA. However, because the ventral pallidum is being inhibited by the nucleus accumbens, the ventral pallidum contains GABA. So you have GABA slowing down something that should slow down the VTA. So because there's excessive inhibition of the ventral pallidum by the nucleus accumbens, this prevents the ventral pallidum from inhibiting the ventral tegmental area. So this ventral tegmental area here is not being inhibited. So the ventral tegmental area is not being inhibited, which means that it's overactive. So the ventral tegmental area contains dopamine neurons, and some of these neurons project the nucleus accumbens, and that's via the mesolimbic dopamine pathway, and others project the prefrontal cortex. So cortical is cortex, and mesolimbic projects to the nucleus accumbens because the nucleus accumbens is part of the limbic system. And these projections can be inhibited through the GABAergic projections from the ventral pallidum. That's what's meant to happen. The VTA is meant to be inhibited by the GABA from the ventral pallidum. So you have the ventral pallidum here that should connect to the ventral tegmental area and inhibit these from firing to the mesolimbic and mesocortical pathway. Because right back to the activity of the hippocampus, so right back to the hippocampus, and that is overactive or hyperactive, that leads through to the nucleus accumbens, to the ventral pallidum, and then now to the ventral tegmental area. And because the ventral tegmental area is overactive, it's sending more dopamine. So there's more dopamine available because it is not being inhibited by the ventral pallidum. So to try and summarize and help understand that, what this blue thing here means is that the blue is glutamatergic. So and this is excitable, so excitatory. And the NAC and the VP here shown in this purple color, or let's call it red, The red is GABAergic, so that's inhibitory. So before here you had the prefrontal cortex and the amygdala, but essentially if we cut that off here, we can say that the hippocampus, the overactivity of the hippocampus creates more, so there's more GABA here because of this glutamatergic projection. Because the nucleus accumbens has GABAergic projection neurons, so that's going to go to here, that is inhibiting more the ventral pallidum. And the ventral pallidum also has GABAergic projection neurons to the ventral tegmental area. So you have an inhibitory, inhibiting an inhibitory. So this is slowing down this. A negative and a negative make a positive, because this is becoming more negative. This excessive inhibition of the ventral pallidum by the nucleus accumbens is preventing the ventral pallidum from inhibiting the ventral tegmental area. And it's quite a hard concept to get your head around, because there's two lots of inhibitory, and because you have two lots of inhibitory and the VTA not being inhibited as much as it was is causing it to be overactive, and the VTA releases dopamine, which is shown in this kind of light bluey color here, and that means that more dopamine is being released because the ventral tegmental area is overactive due to the ventral pallidum not slowing it down. And the reason why it's not slowing it down is because the nucleus accumbens is stopping it from slowing it down by inhibiting the action of it.